All right. Thank you, Dr. Oscarson. And now we're introducing Dr. Veronique Belzil, Assistant Professor of Neuroscience at Mayo Clinic in Jacksonville.
Thank you. All right, I will start sharing my screen. Here we go. So I hope everything is working well. Good morning, everybody. I'm super excited to be here with you this morning. Unfortunately, we can be present in person, but I hope you will enjoy this presentation. It's a very interactive presentation, so if you have any questions during the presentation, please type it on your computer, and I'll be glad to answer all the questions at the very end. So basically, I want to take half of the presentation to try to answer questions that you may have about ALS research, and the second half of the presentation to answer your questions. So obviously the title is Fundamental Questions Regarding ALS Research, and I wanted to go over what is really preclinical research. You are very aware of what clinical trials are, but preclinical research, what is it exactly? So we need to be able to differentiate between basic research, which actually explore questions that are not intended to be used immediately. So basically, people are asking, why is this important? While applied research is more something that has a direct practical application. So something that we can actually use readily from in the clinic. So basically, this is what we are doing for ALS. We are using applied research, and we try to translate what we find in the lab to the clinic as fast as possible. So this is sort of a workflow of how it works. So you have the discovery and development part for the research, and then you have the preclinical research that is readily applicable to the clinical here. And then you have the review process after the clinical trials, and then the marketing of the drug or the treatment. So basically, this is what you are more aware of, I believe, the clinical research or the clinical trials. You have different phases of the clinical trials. You have the first phase where we check if the medication or treatment is safe, and then you look whether or not it is effective. It does the job it's supposed to do in patients. Then you do it in larger scale, if you will, of patients and check against safety. And then when it's marketed, it's on the market available, you look at the effect on the larger population on a long time basis. So basically, what we are talking about today is the preclinical research and how you can contribute to this and why this is important. So first, who can participate to preclinical research? So all ALS patients can participate to these clinical research, preclinical research, all patients with a confirmed diagnosis of ALS. These patients can have a known mutation or not. It doesn't really matter if you know if you're carrying a mutation or not. You can also enroll if you're a spouse or a family member. How it works is that you can have a family member who's a blood relative with ALS or FTD. You know how FTD is highly related as well to ALS. So blood relatives that are affected as well can participate to these studies. We can also have blood relative carrying the family mutation. So if you know which mutation is causing the disease in your family, you can be a carrier without having symptoms. So basically, you are an at-risk person, or we call it a pre-symptomatic person. So these people can also enroll in the studies. You can also have blood relatives that are not affected, and they don't carry, we don't know the mutation that is actually causing the disease in the family. So basically, if we don't know what's causing the disease in the family, and you are a blood relative, you are considered an at-risk member. So you can also participate to studies. And you can also have blood relatives that are not carrying the mutation that causing the disease in the family. So basically, if the family carries a mutation that is causing ALS, but you know that you're not a carrier of the mutation, you're the best control. So for our studies, so you are treated as a control, or just like a non-blood relative would be a control. We can also enroll population controls without any family history of ALS, obviously, but other neurodegenerative diseases. to compare our results to. So the third question would be why you should participate to these preclinical studies. Why is this important to us that you participate to this? Different things. So this is a busy slide. I'll go over it slowly. So we used to see treatment for different diseases as a one-size-fits-all medicine before, right? So we had a disease-specific disease, and then we had a therapy. The therapy was actually working and beneficial for certain people. There was no benefit for for other people. It can be actually worse for some people. So there are several adverse effects. So what we are moving towards now is a more stratified medicine or precision medicine. So basically what it means is that stratified medicine means you will be grouping patients to increase your chance of seeing an effect of the drug, because everybody's different, right? But we can sort of subclassify groups of patients to be able to see a better effect of treatment. So this is what stratified medicine is. And we stratify a group of patients using different criteria, including disease subtypes, risk profile, demographics, socioeconomic factors, clinical features, biomarkers, which are biological markers, and different molecular subpopulations. So we will go over this one very soon. And in terms of precision medicine, how it works, basically you look at the different, you look at the genome of the patient. Basically, we're looking closely at your DNA, and this way we are able to specifically see what would work for you. So this is precision medicine or personalized medicine. This is where medicine is going towards to right now. And we are very excited about the results that we are getting. So this is something that will be established, in the future as a treatment of several diseases. So basically, you can target each patient and see a benefit from each individualized treatment. So basically, we are promoting individualized medicine to quickly impact the clinical practice. So the two important things that we are looking at is, like I said, biological markers. These biological markers are super important to facilitate early diagnosis and confirm diagnosis, but also to confirm how the disease will progress, and to be able to group the patients, like I said before, and measure the effect of a treatment, and also see whether or not a target is working, is actually doing what it's supposed to do. And we also use that precision medicine to develop therapeutic treatments. We target specific drug or small molecules, or we use gene therapy to do that. So I have very few minutes. I'll go very quickly to the fourth question, which is, how can I participate to clinical studies? So basically, you need to meet with your medical team, so your physicians, your nurse, and the research coordinators who are responsible for explaining to you the different research and how you can participate. And then you enroll in these preclinical studies after understanding well what these studies are. And then what they do, they collect samples. So we can collect different types of samples. We can collect blood. We can collect CSF. We can collect saliva by either actually spitting on the tube, in a tube, or just doing swabs. We can collect urine. We can also collect hair. This is highly useful to see if you were exposed to certain heavy metals. And then we can also collect skin. So we do skin biopsies. This is all done in the clinic, and these samples are quickly, within minutes, sent to the labs, and we can process them. So basically what we do with these samples, right after they are collected in the clinic, we can extract DNA, we can extract RNA, we can extract protein, and we can look at the cells that were collected. So basically the DNA is super important. This is sort of your recipe for all the proteins that are produced in your body in different type of cells. The RNA is a copy of the DNA that actually brings the copy to the manufacturer, if you will, the factory, to build these proteins that are sort of the base, the working blocks of your cells that makes your cell healthy and functioning well. So from the DNA, we can look at your DNA sequence and see if you have any variation in your sequence that can either cause the disease or can make you more at risk of developing a disease. And then in terms of the RNA, we basically make a copy of the DNA. And this is sort of a shorter version of the DNA. So you cut it, and you can cut the copy in different ways, leading to different proteins. So we can study closely the RNA and how you copy your DNA at the RNA level and how you cut it will actually define your protein, but will also give identity to your cell. So that's why we have so many cells in our body carrying the same genetic material, but the cells look different and they have different functions because it differs in terms of what we are expressing at the RNA level. And then in terms of the protein, you can have modification of your protein. So let's say it's just a little cartoon to show you the differences, but you have different protein structure here. You can have a closed version versus an open version. So this is the closed versus open is actually caused by modification at a protein level. So based on that, we actually looked at how the cell survives or is healthy or is dying. So cells can actually encounter injuries after these changes, and they just die on their own after the injury, or they can just receive a message to say, okay, this is time to just die. And we call it apoptosis. So it's just sort of a cell suicide signal. So we try to look at these as well. So basically, this is what we're looking at in the lab, and we're trying to understand what is causing the biological misfunction, if you will, in your body in specific subtypes of cells. So what's next? What we do with that kind of information? So we can do in vitro studies. We call it in vitro when it's in a dish, right? So we can use the skin biopsies I mentioned before that you are donating. We can also use cells in the blood. We can use different cells also in the spinal cord. So basically different cells can be grown into stem cells, and if you will. So they sort of return back in time as a stem cell, and then we can reprogram these cells into different subtypes of cells. So we can do neurons, we can do microglia, we can do astrocytes, different brain cells that are very important, and we can look individually at the role of these cells in disease. And then we can edit the genome, your DNA, and reintroduce it to your own cells to see what the effect is. So then when we find something interesting, we can test it in vivo. That means we test it in an animal model. So we have very simple animal models like the worm, like the fruit fly, or the zebrafish that have genes very similar to the human. So either we change the genes that are within these species that is comparable to what we have found in humans, or we introduce a human gene into these animal models to see the effect. We can move on to more complex organisms, just like mice or rats. We can go even further and use more complex animals, just like dogs. I mentioned dogs because there's a disease called degenerative myelopathy in dogs, and it is highly similar to ALS. So these dogs actually, oops, I cropped one leg of the dog, not on purpose. So basically, these dogs have a disease very similar to ALS. So it's a naturally occurring disease in the dog. So we're not causing the disease by introducing a human mutation in these, like we do in these animals. So it happens on its own. And these dogs actually all of them have a mutation in SOD1, which is the first gene that was actually found mutated in ALS. So that's very, very interesting. So it's a naturally occurring disease that we can actually test in the dogs, and we can test the, preclinically or preclinical trial, the treatment we would like to try in humans in these dogs and try to treat the dogs as well. And then we move on to clinical trials. So, and then we can test on a larger scale if these drugs that we found, these targets are safe and are effective at either stopping degeneration or preventing it. So I believe this is it. I will take your questions if you have any. Oh, there's so many. Hold on. Would there be any trials that ALS patients will with low respiratory function can be part of? I believe there would be a question for Dr. Oscarson. So I am aware of all the clinical trials for the preclinical studies, but not the clinical trials per se. So this is definitely a question for Dr. Oscarson or a question for the research coordinators. What else? Family members of ALS patients can contribute blood and or spinal fluid, correct? Yes, they do, definitely. And this is what we actually use the most, blood, because we can extract so many things from blood. And CSF is very, very useful as well, because this is basically what the brain and the spinal cord is bathing with, right? So it has direct contact with this central nervous system that is affected in disease. So this is very precious, but this is what we use most of the time, blood and CSF. How do I get a genetic factor test? So how we proceed usually is when you enroll in a study, we automatically screen for the known genes. So if you want to know if you are mutated or not, and the genes are known to cause ALS, your physician will let you know if you want to know if you are positive or negative. If you don't want to enroll in studies, you can actually go to a private company that will do that for you. So you can check whether or not you are mutated in the known genes. But again, I mean, 83% of cases don't have a known genetic mutation. So this is still a very small number of patients that can be diagnosed with a specific mutation. My family has donated blood through University of Miami. Is this same program? So Michael Benatar is actually leading that kind of research in the University of Miami. So I mean, we're all working the same kind of organizations and steps, if you will. So if you are donating to Miami, the samples are at Miami, but we are part of the collaborating system, if you will. So we are sharing samples. So this is the same kind of approach that we are using across different sites in the US. How do I register to give blood for trials? You just need to get in touch with research coordinators at Mayo Clinic, and you will enroll you, no problem. And we always need controls. So please, if you are control individuals, this is what we lack actually. Patients love to participate, but we need controls to compare results that we obtain from patients. So this is very important. I believe I don't see any other questions. I don't think, do ALS get results about the generic results? I have no idea. Maybe it's a question for Michael Benatar. I think he will be presenting shortly. So maybe we can ask this. I think this is it. I think I am done. I will stop broadcasting and pass the baton to the next speaker. Hope you'll all have a great day, and thank you for your attention.